Okay, I think we're all set. Hi everyone. I'm Kaivalya. I'm one of the managing editors here at Cactus. And today I'll be taking you through an experiential edit of a biomed manuscript. The idea is just to sort of give you guys an insight into, what we do when we're editing manuscripts for different clients around the world. So let me just share my screen. And before we begin, I would just like to give you a short introduction to what we do. So I hope everybody can see my screen. Let me just go to full screen view. Um, a little bit about Editage or Cactus first. Editage is a brand under CACTUS Communications. At Editage, we basically, sort of bridge the gap between journals and researchers. We offer a bunch of solutions. The first and foremost solution is language editing services. And our aim is just to expedite publication for our clients, or at least prevent, rejections on the basis of language. So besides language editing, we do provide other services. So we help under different, teams. We help with things like artwork creation. We help with, clients writing covering letters and things like that. Essentially, we just want that entire, submission and publication cycle to be smooth. So Cactus has a lot of other brands under it that do different things, all obviously aimed at, sort of making the researcher's life better. So you can see the list of brands at the bottom. So we have brands like Impact Science. They work on, communication of science through different, non-scientific channels. For example, websites or magazines and things like that. We have Cactus Life Sciences that works, as it's a medical communications team. Cactus Labs and Unzilo, they're actually our tech branches. So we are developing a lot of tech solutions to help clients as well. And of course, Researcher for Life, which basically helps, researchers with different aspects of their entire journey. A bit about me. So I am, like I said, the managing editor for one of the centers of excellence here at Cactus. So my team basically looks at the subject areas of biochemistry, genetics, and molecular biology. I do have a PhD and a postdoc in the same fields. That sort of gave me a bit of experience with writing manuscripts, with the journal publication cycle. And obviously, I've been working with Editage for the last 4 years, where I've also, sort of worked on quite a few manuscripts across various fields of life sciences. So currently, our team that looks at biochemistry, genetics, and molecular biology, it has over 200 editors and reviewers from around the world. So we're a large group of people that essentially work in our team. That's a little bit about me. I'll move on to the main topic of today's session. Today we'll be talking about, how do we as an editing company or as an editor look at the manuscripts that we receive from clients, and what are the things that we're looking at. Because here at Cactus, we don't just edit, for language and grammar, but we have sort of subject area experts working on these manuscripts, because we basically also want to look at, the presentation of the manuscript. Is it clear, or is the story compelling? I'll talk a little bit about that. So crafting your research story. So the first thing about, manuscript writing is, what sort of an impact it creates on the reader. Obviously, it has to engage a reader's attention. It has to be clear, and it should essentially, get your message across. So that's something that's very important, that we, something that we look at when we're looking at a manuscript. So a few words here. So meaty. So yeah, the first thing that we look at is if the manuscript actually has, relevant information. If all of the results are, sort of supported by enough data, and things like that. So yeah, a manuscript should have, enough information to convince somebody of the study. Of course, it should be clear, and that's quite important. So it should be written in sort of a simple, direct language, so that readers around the world, wherever they're from, can sort of read it without having to sort of figure it out too much. Precise and efficient. This is also very clear. So. How directly are you telling the story? Are you being precise with your descriptions? And efficient, in terms of a manuscript, basically, talks about how the information is presented, right? Is it in an efficient manner, in the sense that, is the reader going from, a broad sort of understanding of your background, of your study, and then narrowing down to your precise results. And yeah, is the information presented really necessary? That's something that goes along with efficient and precise. A lot of the times, we want to put in as much background as we possibly can, or discuss the results a lot, and sometimes that can sort of deviate even from the message that we're trying to send. So yeah, the essential information should be retained, and nothing sort of superfluous. And yes, the delivery of the manuscript, the structure, the way it's written, needs to be effective. So yeah, these are some of the broad things that we look at when we're looking at a manuscript. And I will basically take you through one of these manuscripts. Obviously, along with this, we also look at language and clarity at a sentence level. So let me just move to sharing the actual manuscript with you. Just give me a second. Yes. So I hope everybody can see this screen now. Um, Great. So, this is a manuscript that's been published already, and I just want to sort of take you through how, what sort of changes we make during the edit, and what sort of issues we look at. I've already gone and pre-edited the manuscript, so you can see that I've used the track changes features. You can see a lot of changes that we've made, and we will go through some of them, obviously not all of them. And we will share the manuscript with you separately, so that you can actually go through all the changes. Some of the important ones, that might be handy, have been sort of marked with comments as well, and we'll go through some of those. Um, so yes, most importantly, before you begin, the first thing that we do when we're trying to edit a paper, obviously, if you're not familiar with the subject area, or it's not our study, so we don't really know what's in there. So we try to get an idea of the overall, content of the manuscript. So we scan the paper once. We look at what different sections there are. We look at what, the idea or the main topic of the study is. We might read a few sections just to get an idea of, what the study is about. But what we're also looking at when we're scanning the paper is, just broadly, is the information presented well. We look at sort of like the lengths of the introduction, discussion, just check what's in there. And we kind of make mental notes about, if we might want to look into something. And the second thing is definitely when you're starting to re-edit or edit a manuscript, is to figure out what the aim is of that edit. Do you want to sort of focus on something specific, like word count reduction? A lot of journals have guidelines, and if the manuscript's longer, then maybe you'd want to focus on that. Maybe you want to focus on clarity or improving like concision. You could say, I want to check for consistency. Have I consistently used the same term throughout? Things like that. So, a checklist, if it's your own paper that you're, sort of re-editing or proofing, a checklist like this of what you want to address can be very helpful. If you are aiming for a particular journal, yeah, one should also have the journal guidelines in mind. So, especially the bigger ones that can affect the edit or be addressed along with the edit. So, for example, what's the journal word count targets for each section? Does the journal have any stylistic conventions? So, there will be journals who say, use only passive or use the first person. Journals will have rules about how to use abbreviations, how to use units, things like that. So just having that checklist as well, and not doing it separately, can be helpful. And the final thing is, of course, the edit is not a one-time process. So we do edit in rounds, and that's something one should sort of be prepared for, because you can't address everything in one round. So usually, our first edit is a very basic edit for language. We fix all the very obvious errors, and then we go back over the manuscript to, sort of make the enhancements. That's where you start thinking about, can the sentence be written in a more clear manner? Can you fuse sentences together to improve readability and things like that? So yeah, that's the general process that we go through. So essentially, what I'm going to do is take you through some of these changes that I've made, and hopefully, I can also, go over some things that will help everyone with their own manuscript writing or editing. Cool. So, just looking at it in simple markup. So, like I said, the first thing to look at is, sort of the structure of what there is. So, for example, if I were to look at the abstract, the first thing I would want to make sure is that the abstract, actually serves the purpose of an abstract. So, it's something that should be a standalone section of the manuscript that, readers just read through. They should understand what the study is about. So, yeah, there's a few things that an abstract should have. For example, it should sort of give us the rationale of, what the background in brief is of the study, what the gap is that the study is trying to address. And yes, it should have some of the main methods highlighted, the main results, and obviously it should talk about the implications of the study at the end. Of course, this structure is, journal dependent. Some journals sort of want methods, some don't. But in, broadly, this is what an abstract should have. So yeah, that's what I would be looking for to begin with. Does it have all of this? So, for example, you can look at what I've underlined here. In this particular manuscript, the authors basically say, they're looking at this protein DPP-A3, and they say it plays a critical role in the regulation of DNA methylation. And then the next sentence says, however, the role in protein stability remains unclear. Now, this is fine because essentially you've told the reader what it already does and what is unclear. But something that maybe I would suggest, to the author to add here is sort of why. So if DBP3A is known to regulate DNA methylation, why suddenly are we looking at its role in protein stability, right? That's a completely different process. So there's no link essentially to really tell the reader why this particular question. Why are we not looking at its role in, say, another process in the cell? So yeah, that's a little bit of an information gap. Where someone might just say, okay, but why did they think of this? So I'd recommend maybe adding that. And yeah, I'd look at the methods, the results to see, okay, is enough explained. And finally, I think the most important bit is the conclusion. It creates an impact on the reader. So I would want to see, whether they've actually told the reader their main finding and how it actually influences or creates what's the significance of that finding, right? So they have said, it helps us understand the role of DPP3A more, which is fine. So that's the broad sort of check that I do. And similarly, like we do this for all different sections. So even in the introduction, I would check, is there, enough information given for me to understand, what DPP3A does, why are they looking at its, sort of role in protein stability, things like that. And does the introduction actually convince me of, that there is a rationale to this study? So yes, first checking the structure and the information presented in every section. Then, of course, there are edits that are made at single sentence levels, and for various reasons. So maybe I will go through some of these. Um, so right at the start, to begin with, we've made a change over here. So the clients used, ES cells originally for embryonic stem cells. Now, in literature, this is quite commonly, referred to as ESCs. Um, so instead of saying ES cells, we've actually gone ahead and replaced this. Now, embryonic stem cells are used, are the basic topic of their study and they're used throughout. So immediately, it makes it easier for the reader because, you're introducing just one abbreviation. That's one thing. But the other thing that it does, especially if you're looking for word count reduction, is that it cuts two words into one. And yeah, this is actually a good way of sort of making sure your word count doesn't increase too much. If you are using terms again and again through the manuscript, think about whether you could introduce abbreviations for them. So yes, in this case, we've sort of done it because it is a commonly known abbreviation. And we also noticed that the client has used PGCs for primordium germ cells. So if you are using, C for cells here, then why not here? So just for that consistency as well, we just sort of, suggested to do that. That's a sort of change that we would make. Then again, we have fix some phrasing issues. Sorry. So, for example, the original said, it plays a critical role in the regulating of DNA methylation, which is just essentially a grammatical error. So it should be the regulation of, and that's what we've done. Now, sometimes we also fill in sort of information gaps if that information is available, if it's common knowledge or if it's available in the manuscript. And this is something that maybe even if you're writing a manuscript could be helpful. So, for example, here what we've done is we've added some information on one of the methods. So what the author originally basically said is that he just said, after Go analysis, we further constructed blah, blah. Now, Go analysis is, maybe common knowledge for those who work in that field, in sort of the, pure biochemistry field, and do sort of protein network analyses and things like that. But not everyone really knows what that does, or why you would do a Go analysis or a gene ontology analysis. So what we've done is we've basically added that information, right? So instead of just saying after Go analysis, we did XYZ, we basically added the functions of the interactors were analyzed using gene ontology analysis. So this sort of missing information that just tells the reader, what the purpose of this analysis was, is something that we look for these gaps in information and add them if possible. Yeah, so another phrasing error that I want to bring up, and I think, and I've seen this quite a lot in manuscripts. So I think it's a common error and it's a difficult of sort of phrasing. So mechanisms of X on Y is very commonly used, which is sort of incorrect. So for example, here. Also, the original was, to explore the mechanisms of DPP3A on protein stability. If you read it fast, it sounds okay. But it technically doesn't make any sense that, you're talking about the mechanisms of something on something. So the mechanism is actually the underlying process, right, of how one thing has an effect on the other. So I think it's a common thing that people struggle with how to phrase these, and underlying is a great word to use with mechanisms. And yeah, and even in this manuscript, this has been repeated several times, mechanisms of in incorrect manners, and I've changed that everywhere. So yeah, a good way to tackle mechanism related sentences is to just go with underlying. So you essentially say mechanism underlying the effect of X on Y. So yeah, that's one common error that I thought would be helpful to talk about. Um, Another thing that I feel can be quite helpful when you're editing or writing is thinking about concision. So the more concise you can be, the better it is, just in terms of readability, right? So one way to ensure concision is just to find words, single words that can replace longer phrases. So, for example, here we've replaced quite a few with multiple. Yes, we've obviously, in terms of word count reduction, gone from 3 words to 1 word, but also it just makes the sentence nicer to read. A bit more concise, a bit more precise. And there are many examples like this. So, for example, a lot of the times all over the world is used quite often. And that can be replaced, for example, with globally or worldwide, words like that. In addition to or in addition can be replaced by additionally. So, yes, there are quite a few words that you can actually replace with. They're smaller. They're shorter versions. And in relation to this is another example. A lot of the times we use, opposing or contradictions. So, for example, we will say something like, we found that the size of X was not larger than the size of Y. Now, this not larger, can just be replaced by smaller. And we do this quite often in scientific manuscripts where we say something is not X rather than using the actual word. So, not larger, not faster instead of saying slower. And that's another good thing to look for if you're aiming for concision. Um, yeah, I have a couple of other examples. This one is really helpful as well. So whenever I have to reduce word count, especially if I have to like forcibly do it to meet a journal target, I will usually look for these phrases. So phrases like X of Y. So for example, here degradation of nanoprotein. You can always flip these to sort of remove the off. So instead of saying degradation of protein, you can always say protein degradation. And similarly, a lot of things like this, right? Expression of genes, say, which can be turned into gene expression, growth of cells, proliferation of cells. All of these can be cell growth, cell proliferation, things like that. So I actually look for phrases with off in them and see how many of them I can sort of flip around to just get rid of the off. That's a great way to do this as well. So here's sort of another example. Here it says, stability of proteins. You can just change that to protein stability. Um, yeah, so a few ways there of making things shorter. Another tip that I have, you can see here again that we've made that change to embryonic stem cells from ES cells to ESC. Now, like I said, this is used throughout the manuscript, so there's really tens, or hundreds of mentions of this throughout. Now, how do you make sure that you're using this throughout consistently, and then you don't by mistake switch back to embryonic stem cells or by mistake switch back to ES cells? Or if you've decided to make this abbreviation later after you've written, how do you make sure that you catch every instance? So control F checks are sort of one of our favorite things to use because they really help with catching all instances, right? So you can use control F, which is the search feature in Word. And basically you want to search for, all instances of something that you want to replace. So for example, here, I basically searched for ES cells. And wherever that phrase appeared, I replaced with ESCs. Um, and essentially you can, instead of doing control F, if you want to do it a bit faster, you can use control H, which is basically find and replace. So if you use control H, basically Word gives you the option to find something and replace that with something. So you can basically find ES cells, and in replace you can tell Word to just put ESCs. And you can either replace all together if you're feeling brave, or you can even with a single click go to go to each instance and change things. So control Fs are a great way to ensure consistency. We don't usually rely on finding it by reading through the manuscript, because that can be it's prone to errors. Another thing that is helpful to remember is the rule about abbreviations. Now, abbreviations really do help sort of make things concise, but um, they're not really required if a term is not repeated too many times. So the general rule is that if you use a term less than 3 times, don't use the abbreviation, because whenever you introduce an abbreviation, someone has to retain that, right? So when I read inner cell mass and ICM, I have to remember that ICM stands for this. So if ICM is only being mentioned somewhere once again in the manuscript and not too many times, then I probably won't remember what it is, and I'll have to go back to check what it was. So unless it's a repetitive term, usually using abbreviations doesn't help the reader. And sometimes we also feel like maybe they are like slightly more common abbreviations and things like that we just introduce. But essentially, if you're just using the term a few times, basically that means less than 3 times, it can sort of hamper readability. So that's something we do check for when we edit. So we check how many times a particular abbreviation is used, and if it's used less than 3 times, then we essentially remove it. Yeah. Okay. Another important rule, is tense usage. So this is again something I think that's a bit confusing for people, and people sort of don't always get it right, or it's inconsistent. So with tense usage, a good rule of thumb to follow is that if you're reporting something that was already published in the past, and you're using it to basically say, this is like a fact, right? So you're presenting it as your background information. So in those cases, you can actually use the present tense because it's being sort of stated as fact. An example here is to say DPP3A, whatever, mutant male mice showed normal fertility when mated with this and this. But what the client is basically talking about is a finding of a previous study. So instead of saying showed, you can actually just change it to show, because what you're trying to do here by presenting it in the information is stating it as a sort of a fact or something that has already been shown. So you can go with the present tense. So it's sort of like when you state other facts, like the sun rises in the east, you use the present tense. So you can do that. An exception to this, could be if you say, if you use the author name. So let's say you say, I don't know, Young et al. Showed that. In that case, obviously, you have to use the past tense because you're talking specifically about that particular study or the particular author showing something. But if you're just telling about the findings, you can use the present tense. And that's throughout the introduction, more or less, when we're just talking about background information. Great. So pardon the big gaps, or this is just a function of going into all markup. Here's another example of the mechanism thing. So again, here, the mechanism of DPP3A on DNA methylation is incorrect. What the authors are trying to say is the mechanism by which DPP3A affects DNA methylation. In this case, instead of saying underlying, we could have done that. We could have said the mechanism underlying the effect of DPP3 on DNA methylation, or just say, the effect of DPP3 on DNA methylation. Another example of that. Okay. Here is an example of, again, sort of, the phrasing not being clear enough. And these are, maybe small things that we miss out on especially when we're writing our own paper, because we know so much already about it. So we tend to, miss out on small pieces of information. So the sentence here basically said a defined medium known as 2I medium. And then it basically had, in brackets, it basically said MEK inhibitor this and GSK inhibitor this. And then the rest of the sentence, right, contributes to the establishment of, etc. Now, for somebody who doesn't know what this 2I medium is, obviously they've coined the term because they created it in maybe for the study in their lab. Um, 2I medium and in brackets, just having the two inhibitors, it doesn't really tell us much. It doesn't tell us, are these inhibitors sort of part of the medium? Um, why are they mentioned? Is that all the medium is comprised of? So things like that. So that's not like a very clear way of presenting it. So yeah, what we've done is basically, added this phrase that says, to a medium that contains a MEK inhibitor and a GSK inhibitor. So that tells the reader exactly what these inhibitors are doing in that medium. So small things like this, where in our labs and when we are working, it's so common knowledge to us that we don't think maybe it's unclear to a reader. And yeah, so things like this are what you could check in your second or third rounds of checks if you've written that manuscript yourself. Um, Great. Another example that I want to talk about in terms of just making the manuscript clearer or more concise is combining sentences. Um, a lot of the times we sort of break up sentences into multiple parts, and that can be helpful. If your sentence is very long, has too many commas, or presents too many ideas, then it's not a good idea to have one big sentence. In those cases, yes, breaking it up into smaller sentences can help readability. But if your sentences are essentially repeating information, and yet they are broken up into different sentences, there it can really work to combine them. So, for example, here we've got two sentences. One is specifically we found that the overexpression of full-length DPPA resulted in the accumulation of this UHRF1 protein. And then the second sentence is, furthermore, DPP3A overexpression increased NANOG in ESC. So essentially, there are two sentences giving you similar information, where in the first sentence you say DPP3A overexpression leads to accumulation of UHRF, and the second sentence says increased NANOG. So what we've essentially suggested is to club these sentences together and just say we found that the overexpression of DPP3A resulted in the accumulation of UHRF1 and NANOG. So there's actually no reason for the sentence to be broken up into two pieces when you can just combine and present it. Right? So yeah, obviously at the end of the introduction, yes, we check for structure of the introduction again. Like I said, is enough background given? Are the gaps and rationale clear? And most importantly, does it end with aims and implications? So most journals do not allow us to, summarize our results at the end of the introduction. Some do. But in most cases, they want us to end with like a aim. And if you can, what would the findings sort of help with? So that's what introductions should end with. Yeah, we would also sort of say don't reveal the results at this point, but rather just talk about what the aim is. So we aimed to explore the role of DPP3A in protein stability by looking at UHRF and NANOG expression or things like that. So even this reveal of what it does, we would sort of discourage. And then of course the implications. So these findings will provide new insights into X, Y, and Z. So yeah, a good introduction should ideally end with convincing us that the results will have an impact. Great. So I'll move into the materials and methods. There's not much that I will sort of discuss here, but a couple of tips that we usually come across that might be really helpful. One is how manufacturer information is presented in the methods. There's usually a lot of inconsistency in terms of this. So you can also see here it says GIPCO Thermo Fisher Scientific. What we've deleted here says GIPCO Brazil. Here it says GIPCO USA. Um, so yeah, it's all sorts of things. So it's very important to check if the journal has a requirement. So a lot of journals have requirements specifically for manufacturers in the US, and not so much for the others. So for example here, this journal basically told us that if the vendor is in the US, you have the state, company name, city, state, or country. And if it's outside the US, you can just sort of give us the company name, city, and country. So it's important to check this. And then it's important to go through each instance to make sure that that is consistent. So this is one of the number one problems we come across in materials and methods. Another big another often repeated issue is consistency between units. So materials and methods is somewhere where units are repeated, throughout. You're describing buffers, temperatures, times, all of that. And while you're typing, it's very easy to use different sort of styles for units. So, sometimes you'll use a space before the unit. Sometimes you'll forget and, just say 2 mm. And things like that. Another very common one is how ml is written. So SI units require you to write it with a capital L, but obviously a lot of times in lab or when we're being informal, we would use the small lowercase l, and that sometimes happens when you type. That you would just use the lowercase l, and it's happened right here where we've corrected it. So in this case, the ml was written with a capital L, and here it was written with a small l that we changed. Now this is very, very common, and again, it's not a great idea to just check with your eyes. So control F's come in handy again over here. So I always look at the units that are used, and I do a control F for the units, and I will search for m lowercase l, and whatever instances I find, I will replace them at once with the right version. Yeah, you can also search for spacing issues, and a good way to do that is using wildcards on MS Word. So if you just Google this, you'll get a list of the wildcards you can use. So in your search bar, if you're looking for, say, example, I want to search for something like this, where there's no space between the number and the unit. So what I would essentially be searching for is that combination, or a combination of a number followed by a letter without a space in between. And wildcards actually allows you to do that. So in your control F in the search bar, if you put in you know the symbol that I have forgotten what it's called, but yeah, that with a hashtag. So that basically, this bit basically tells Word that you're looking for any number. And the $sign tells Word to look for letter. So you're basically looking for any number followed by any letter, right? And there's no space in between. So you're looking for things without a space. And when you search for this, Word will essentially find all combinations like this for you. And there won't be a lot of combinations. So if you search through the materials and methods, you will most likely come across all the instances of numbers followed by letter without a space. So it's a good way to search and fix it throughout. So yeah, a lot of materials and methods is about consistency and making sure that all the information is presented. Um, yeah, another sort of tip is to check the voice, and you stick to one style. So oftentimes we switch between active and passive. So we say, we did this, and then this was done, and then you go back to we, and that's just not easy to read. So yeah, one thing is to figure out if your journal has a requirement, and if not, which version you'd like to go with, active or passive, and sort of stick to that throughout. And these are sort of the changes that we make as well for consistency. Yeah, so I've also shown obviously a lot of phrasing issues through the materials and methods that I don't want to go through. And when I share the file, you guys can look through this because they're very specific to this paper. Yeah. So we will maybe take a couple of examples where logic, like I showed before about the 2I medium, how is the information like sort of making sense? Are we just sort of using colloquial language that we might use inside a lab? These are a couple of very often repeated examples when it comes to things like Western blots and things like that. Um, where we simply like here the author has simply written you know yeah so using 5% skim milk to block signals non-specifically for around 2 hours. Yeah, to block signals non-specifically has a very different meaning from what milk actually does in a Western blot, where it actually, blocks non-specific signals and doesn't block signals non-specifically. So that's a very different thing. So yeah, just keeping an eye on things like that, where we changed it to say something like non-specific signals were blocked by incubating the membranes in milk. So these are very nuanced things, and that's where you need sort of a subject area or person to work on the manuscripts. Um, and things like this, where you say, an antibody was added and incubated overnight. So what was incubated? So here as well it says that primary antibodies were incubated overnight at 4 degrees, which is completely incorrect. So what you're doing is incubating something with that antibody overnight, and that is the kind of information we've added here. The membranes were incubated with the primary antibody overnight. So yes, again, very, very common, sort of, uh, phrasing issues that we look for. Um, yes, again, like I said, all the information that is required should be present. So a lot of the times catalog numbers for antibodies are really important because they, batch to batch have variations. So that's something that we look out for if that's added. Um, Great. Okay, so some more examples that I think you guys can go through later when we share the manuscript. But yeah, I basically pointed out, other ways of, again, combining sentences, making things more concise. Um, and again, clarity similar to the one above to make sure that the information is accurate or the method is accurately presented. Now, what we definitely do when we're going through the methods, and one should do essentially, is make sure that every method has a place in the results, and vice versa. So essentially, there shouldn't be any methods that are not eventually used to describe one of the results. That can sometimes happen because we, maybe don't in the results put in everything that we've done. We obviously pick and choose the experiments we want to showcase. And sometimes we can end up, forgetting to remove that from the method section. So every single method should definitely have obviously a place in the results and vice versa. And so what we do check is, everything that's described in the results, is there a corresponding method to it? Okay, so I'll give you an example here. The clients did say that they did go term analysis or a gene ontology analysis. But how they've done that is not described in the methods at all. So that's something that would be critical because this is how they basically identify proteins they want to work with. That information is missing from the methods. There are other cases like that where we found that they haven't supported whatever they've written here with the methods either. um, cool. Yeah, I will maybe skip a few of these examples and yes, show you the other example of missing information. So this is a small thing, but they've suddenly introduced co-IP or co-immunoprecipitation. Nothing of that was ever described up in the methods. It's a big procedure, so we would want to, have a section on this in the methods as well. Yeah. So this is a really important check that you do with the results. Again, a lot of concision examples. I think this is another example of where we've gotten rid of off. And instead of saying regulation of DNA methylation, we've just said DNA methylation or regulatory mechanisms. Um, yeah, we can also remove a lot of these words. We oftentimes say things like it was reported and the PP3 is reported to regulate and things like that. And we don't really have to say it. We also don't have to say things like, X, Y, and Z at all showed because you're putting a citation at the end. So that's a great way to just get rid of extra words as well. Again, like I said, just present things as fact instead of saying is reported to regulate. Just say DBP3 regulates methylation level and have a citation. So that is also reported to is covered with the citation. So yeah, as much as possible, it's best to just present things as sort of facts or previously proven findings, rather than trying to say in that study or authors of this study showed that or things like that, which is not really necessary um, to drive the point. Cool. Again, since we're in the discussion, we're also looking at how the discussion, the information is presented. So now, ideally, the discussion should focus mostly on what the results mean in terms of other literature, right? So a lot of times, what we tend to do is kind of recap the results in the discussion. It's not necessary because the reader can just sort of go back and look at the results. So by the time they've got into the discussion, we sort of expect them to kind of know the results. So yeah, these kind of statements like, our data shows that DPP3 interacts with X, Y, and Z, and this, and all the alpha subunits, etc., etc. It's not required in the discussion. So we can just, something like this should be part of the results, even if it's a supplementary information file. Yeah, just to interrupt the discussion, by repeating results is a bad idea. Um, yeah, a good check or to sort of check if you've done that is, I mean, what we do is look for figure citations that essentially tells us that there's something to do with results that's obviously put in the discussion. Of course, if it has some mechanism or flow chart, or that's fine, but usually references back to figures in the discussion is a red flag, because that means they're talking about results. So yeah, avoid repeating results here and just get to the actual comparison with other results. Yeah, cool. Okay, so I think there's a couple of other examples here of clarity. The changes we've made to just make the sentence sound better. We also again have found certain instances where things can be moved around. So this is a good example. You will see that most of this paragraph talks about ubiquitination and protein degradation pathways and things like that. And somewhere in the middle of the paragraph, you have this statement that says protein degradation is vital for removing misfolded and damaged proteins. A, it's a very, very basic sort of statement. So one should think about whether such sentences are required, especially if you're aiming for a very specific like journal. So in this case, stem cell research and therapy. It's not a very broad journal. That the readers will have a certain basic level of knowledge. So yeah, describing very basic things. I think somewhere up in the methods, there was also a section that says, we ran PCR to amplify, certain whatever fragments. And I think that definition of a PCR isn't required for a scientific audience. So just to be wary of presenting very basic information. And if it is being presented, then, to not have it just in the middle or to have it right at the top. So yeah, the idea always is to go from broad to more and more specific. So the broad, like the most general things on top, and then more specific things as you read along. Yes. So yeah, I think again another sentence that's totally out of context here, and I think just inserted because it's another function of the protein. it was just sort of misplaced and didn't really add any information to that discussion. So we also sort of suggested removing it. Um, yeah, so that's sort of the end of the paper. Of course, I didn't go through all the examples, but I've sort of explained quite a lot of the phrasing issues at the very least. So if you're interested, you can go through them later. So what do I do at the very end? This is very important. So of course, a spell check. The word spell check feature is very, very helpful. If you go into spell check settings, you can actually even select what it should look for, in terms of active phrases, passive phrases, or things like that, clarity and concision, and things like that. So it will even flag things that can be made shorter for you. It'll flag, active versus passive, and that can really help as well. So we run a very extensive spell check with essentially all of the settings selected. Um, Again, Control F. This Control F that we run right at the very end is to check for little typographical errors. So double and triple spaces. So as you're typing, it's very like easy to introduce multiple like double spaces, and you won't even realize it. So a Control F where you just hit the space bar twice. It will basically show you all the instances where you have double spaces. Yes, it's a lot right now, but yeah, there. If you go to simple markup, yeah, so it's basically going to take you through each of these instances of double spaces so you can fix it. And go to the next one and fix it. And similarly, you can hit the spacebar thrice, and that's going to show you if there are triple spaces. And then we do a bunch of combinations. So sometimes you can have a double period at the end of a sentence, something like this. And you can quickly go and fix it. Similarly, for all other punctuations, like a double comma, if it's there, there we go, we fix that. Um, yeah, think of what are the possible errors. A very common error is to have a space before a period. So I would search for a space and a period. Um, and that basically gives me instances like this where a additional space is added. So you can think of various different combinations of this. So I don't have a space and a comma error. Um, yeah, just doing these basic like spacing control Fs can prevent all of these small little oversights from remaining. And yes, at the very end, one thing that I find very helpful to do is rechecking the title and the abstract. Essentially, because you've gone over the entire manuscript, you have a better idea of what is presented, what the results are, what the study is. Just going back to do a double check of whether the title is really representative of the manuscript. Can it be made more specific or can it be changed? And same for the abstract as well. Just whether it actually covers, enough of the findings, and things of that sort. So a good idea to do that once you're actually through the manuscript. Of course, if it's your own study, then you don't have this problem. But for when you're maybe reading somebody else's manuscript, or if it's not something that you've written, this is a good check to do. Um, yes, so that's it. That brings me to the end of this. So yeah, we will be sharing this entire edited manuscript where you can actually go through all the changes and understand a little better. Yeah, but that's about it. Thank you. And of course, if anyone has any questions, I would be happy to take them. All right. Yeah, so Aditi shared a feedback form, and it would be very helpful for us if you guys could fill it. And yeah, let us know if this was helpful, if something could be changed, or if you can do things differently, and we'll sort of incorporate that. Great. Then thank you. And I hope this was a bit helpful for you guys, whether you're writing or editing manuscripts. I hope you could take something away from this. And yeah, thanks a lot for attending. Um, and I hope you have a good evening. Thank you. Bye.